we are going to talk about the development of a candidate for reference material from genomic DNA of Salmonella. This work was carried out by the Bioanalysis Working Group by the Instituto Nacional de Metrología de Colombia. Salmonellosis is one of the most common human foodborne diseases produced by members of Salmonella genus. According to WHO, it is estimated that it affects millions of people around the world, especially children and elderly people, causing more than 100,000 deaths per year. Cell culture has been the gold standard method for this identification. However, it can take from 1 to 2 weeks to get conclusive results. In contrast, there are several commercial PCR assays to identify Salmonella spp. Most of them are based on their own internal positive controls, but sometimes there is not a consensus among the different responses due to a lack of common reference to make them comparable. Thus, the objective of this work is to develop a candidate to reference material in terms of genomic DNA as a contribution to the dissemination of traceability and comparability of measurement results related to the identification and quantification of Salmonella spp through PCR. How we did it? With the aim of producing and characterizing a reference material based on a Salmonella genomic DNA, first we validated a droplet digital PCR assay, ddPCR, based on different DNA Salmonella targets. Then we optimized the Salmonella growth kinetics in selective medium and optimized a DNA extraction method. And finally, we produced the candidate reference material. In order to develop a digital PCR method to characterize the DNA from the Salmonella enteritidis reference genome CP00743 and literature, there were selected 6 different regions corresponding to genes INVA, SPAQ, HILA, TTR, SOPB, and CIIA. First, they were evaluated through real-time PCR in simplex and duplex form to evaluate the fitness for purpose. Figure 1. Salmonella enteritidis genome scheme. The genes for the PCR method are shown. They were selected, designed based on a literature review and blast analysis. Based on the response in digital PCR, the 3 genes INVA, HILA, and TTR were selected to validate a digital PCR method. The INVA and the TTR were evaluated in duplex, while HILA was evaluated in simplex form. Taking advantage of the duplex reaction, the DNA characterization was performed with INVA and TTR sequences. The validated digital PCR method has a linear range between 8 and 8,000 copies per microliter in the reaction mix in the duplex form as it is shown in the figure. A detection limit of 2 copies per microliter and quantification limit of 8 corresponding to the lowest level of linear range validated. The precision in the validated interval was between 18% for the lowest concentration level to 3% for the highest one, measured as a relative standard deviation over the evaluated span. The assay specificity was evaluated against 4 Salmonella and 13 related species, obtaining coherent results. The Salmonella strain, preserved at -80 degrees Celsius, was recovered and activated in a nutritive DSA agar. In a selective Rappaport bacillarius broth, the cell culture was scaled up 200 milliliters in order to obtain enough biomass to DNA extraction. A XLD agar was used as a quality control in order to guarantee the identity of the Salmonella strain. The figure presents the cell growth kinetics, counting the colony-forming units and measuring optical density. From there, 10 hours were selected because the stationary phase begins, with an approximate concentration of 1.4 to 8 CFU per milliliter to stop the cell culture growing and to extract the DNA. DNA extraction was performed by a modified CTAP method, where the changes were associated to RNase lithium chloride concentrations. The DNA quality parameters evaluated were DNA contamination with spectrophotometry, where the absorbance 260, 280, and 260 and 230 ratio indicate no contamination with proteins and/or extraction regions. DNA integrity through agarose gel electrophoresis, where lane 1 and 2 contain the DNA extracted, lane 3 negative control, lane 4 DNA base pair ladder of 100 base pair. There is no smearing indicating the DNA is not degraded. The presence of inhibitors was evaluated through real-time PCR, and according to the efficiency obtained for IMVA and TTR sequences, there is not inhibitor presence. After that, the digital PCR method was validated, and the cell culture and DNA extraction methods were optimized. The production of the candidate reference material began. With the DNA stock solution were prepared 6 gravimetric dilution levels in the range of 10 copies per microliter to 200 and 1,000 copies per microliter, which were packed and labeled in 1.2 milliliters polypropylene tubes. After it were performed the DNA solution characterization. From the homogeneity study data, it were evaluated the homogeneity, the reference value, and the uncertainty component associated at homogeneity. After, it was performed a short-term study for 4 months to evaluate the material stability. With this data were established the combined uncertainty and the value assigned. The homogeneity study was performed as follows. From each concentration level were taken 6 samples. They were measured in duplex form with IMVA and TTR sequences 3 times. With the measurements results were obtained the values and combined uncertainty for IMVA and TTR. Using these values, the concentration reference value for each level as well as their uncertainty were obtained. For INVA and TTR sequences, the main uncertainty sources come from the homogeneity and characterization. This one is related with the precision and mathematical model. For the reference value, the bias between IMVA and TTR results is an additional uncertainty source. The stability studies were performed for 16 weeks at 4 Celsius degrees and -20 Celsius degrees. The results showed that the data of the highest concentration levels are closer than the data of the lowest concentration levels. All levels were stable up to 12 weeks to evaluated temperatures. However, the stability of the highest concentration of the material is longer, so currently we continue evaluating it. In addition, a freeze-thaw study was carried out. It allowed to establish the stability of the material is affected by the process. So, to guarantee the assigned value, the material just can be freeze-thaw twice. Every data corresponds to the average of 2 samples. We will continue working in this topic in order to improve this behavior. Regarding to the uncertainty budget, the study allowed to establish that for the highest concentration levels, the main contribution to the uncertainty is the bias between IMVA and TTR targets, while for the lowest one, the mathematical model is the main source, as it is shown in the figure. For the confined uncertainties, the table shows that it is between 2.6% for the highest concentration level and 24.3% for the lowest one. Conclusions. Based on the stability study, the material produced is appropriate to be used as an assay item in a proficiency test. Uncertainty is a concentration function. At low levels prevail the mathematical model. At high levels prevail the bias between genomic sequences used to characterize the material. This genomic DNA candidate to reference material is the first one produced in Colombia. Thank you for your attention. If you have questions, please contact us to the below email address.